infectious diseases have been one of the largest threats of humankind and have even threatened human civilizations, such as the plague in the medieval ages or the recent COVID pandemic. Many of these are not intrinsic to humans, but so-called zoonotic diseases that can jump from the animal hosts to infect humans. These emergent infectious diseases have been exerting an enormous toll on humanity, such as COVID, the HIV pandemic until effective drugs against the virus became available, or the 2014 outbreak of Ebola in Western Africa that has claimed more than 11,000 lives. One response to the threat of zoonotic events are surveillance and monitoring projects in areas where humans, through hunting or by encroaching on pristine habitats, come into close contact with wild animals and thereby at risk from becoming infected. Many of these surveillance projects are situated in Africa. They focus on the zoonotic potential of viruses or bacteria in bats, rodents, monkeys, or apes. I am Holger Breithaupt, Senior Editor at EMBO Reports. Today I'm talking to Fabian Leendertz, Director of the Helmholtz Institute for One Health in Greifswald, Germany. One Health is a collaborative effort between physicians, veterinarians, molecular biologists, ecologists, and other experts to identify emergent infectious diseases so as to protect the health of not just humans, but also animals and the environment. Fabian leads a long-term interdisciplinary surveillance project in the Tyne National Park in the Ivory Coast. Scientists, physicians, and veterinarians systematically study our closest relatives, the great apes, for emerging diseases. The July issue of EMBO Reports features a fascinating article by him and his colleagues with much more detail on their work and what they have found so far. Welcome to the EMBO Podcast. Well, thank you very much for your time first and for the interview and the podcast. You're director of the Helmholtz Institute of One Health. Can you tell us a little bit about the concept of One Health and how the Institute's mission fits into this larger context?
So One Health is a concept which says that you should not work in isolation in different disciplines, but across disciplines. And specifically here, it's the medical disciplines, focus on humans, but also the veterinary disciplines and the animal disciplines, including biodiversity, and then the environment, broad environment, which really includes climate, habitat, and so on. So it's a really all-inclusive concept saying that when you change something, very roughly, if you change something, one of those factors in the environment, you will influence, for example, animal communities, and that again can influence health of people. And that can go in all directions. Here at the Helmholtz Institute for One Health, we're a very new institute. We really have this One Health concept as a foundation. So we try to work equally on the aspect of human health, animal health, biodiversity, and environmental factors, which is a big challenge because it's across various disciplines. We have to learn to speak the same language, to develop the common ideas, concepts, and then put these things into translation, which is something One Health urgently needs, because there are lots of strategy papers about One Health. But if you look at concrete examples on how it can work, even data can be collected or translation can happen in terms of findings, and you will always find the same two or three examples. And so I think we need to provide evidence that One Health can work.
In that regard, prevention and surveillance is a big part of One Health and part of your own research, and one of the bigger and long-term projects of yours is these Thai chimpanzee projects, of which you had the article in EMBO Reports. Can you tell a little bit about the project and how this again fits into this overall view of surveillance and prevention and enabling One Health?
The chimpanzees, especially in Thai, they're like my oldest topic, and that's where I started my scientific pathway in 1999 and starting 2000, and in the forest, focused on chimpanzee health. Because Christoph Bösch, at that time director of the TCP, Thai Chimpanzee Project, said, hey, my chimps are dying or sick, and I don't know why, and that's bad for my behavior research. So I was the first batch to look at these issues, and we discovered a number of pathogens which are of importance for the great apes, but also potentially for humans. And since my lab at that time was at Robert Koch Institute, and Robert Koch Institute is focused on public health, human health, we obviously quickly took the next step to look into the human communities. Do those pathogens transmit from animals to humans? Do they matter for humans? And then from there, very quickly, we realized that the different habitat types are very important for the potential reservoir species, which are part of that puzzle. We also started working already in 2004 on the small mammals like bats and rodents living in different habitat types. So this is where very soon, just by thinking about what is the problem, where does it come from, and what are the main drivers, you run into a One Health concept, at least in the environment where I've been working for a long time.
A central point of the project is that you don't disturb the animals and you do all the sampling in a non-invasive manner without even physical contact with living animals in the forest. That sounds quite a bit of a challenge and a hurdle there if you want to study the pathogens that afflict the chimpanzees.
Yeah, there were some technical hurdles because those animals, they're wild. There's no fence around and they're not fed by us. They're not touched. Because they serve for conservation and behavior, so primatology research. So if we would starting anesthetizing those animals, they would very quickly remember that, and that would disturb our relationship to the animals. So since the main purpose of the project is this primatology research, and we did not want to mess up our access to the animals, so also for our own purpose, we had to invent non-invasive methods. But to be honest, we were not the first to invent the first steps in that regard. Beatrix Hahn, who many people know in this infectious disease world, was the one who discovered that you can find actually SIV in the feces of chimpanzees. This is how she, together with Martin Peterson, pinpointed where the spillover from chimpanzee to human led to the big AIDS pandemic, right? So it's Southern somewhere. And so we said, okay, if that works for a systemic pathogen like HIV, SIV in that case, it should also work, for example, for respiratory diseases, because you swallow what you have in your nose and so on. And indeed, that worked really well. And that allowed us to also look for other pathogens in feces. You can also find antibodies in feces or urine. So that's sort of the next question of past exposure. That works well. However, having said that, for the Thai chimpanzee project, we work non-invasively and we don't want to disturb their behavior. But when they're dead and the others are gone, then you can do a full necropsy. So this is what we are doing since 24 years now, to do full necropsies on all animals found dead in the forest, especially the chimpanzees. And then these are known individuals, right? They have their names. We know them in some cases 20 years. So we have urine and feces sample from that animal before death. So we have a very good blood and liver, spleen, whatever sample, plus the non-invasive samples from before. This allowed us to also set up many of those methods and have a good idea about sensitivity and specificity and these kinds of issues. So it's that type of mix of post-mortem plus non-invasive, which is very useful for us.
And you've discovered quite a number of pathogens there, from monkeypox to human respiratory viruses, leprosy, and so on. And also given that Ebola and COVID also emerged, were zoonotic events most likely from wild animals. So what else do you think is lurking out there as a potential threat that could eventually spill over into humans?
You know, when you go in a nice tropical rainforest or in the disturbed habitats at the edge of the forest, and you start looking for new viruses. I had a PhD student, for example, a long time ago. She was catching mosquitoes and cultured viruses like that to just see what types of viruses are there. You will find many, many new types of viruses you have never thought about. The question is, do they matter? They're interesting. I'm not saying they're not interesting from a taxonomic point of view and so on, but do they matter for mammals infected by them? Right? And so this is where we have the great apes as our closest living relatives. They live out in the jungle. They live there naturally all the time. They sleep there, they eat there, and so on. They have their families. And so when they get sick or die, whatever killed this animal or made it sick will also be highly relevant for us. That people used chimpanzees in the old times. Luckily, that's over, mainly in the US, to look at the impact of certain pathogens on humans. They were the perfect model animal, right? But we have that model running around wild in the forest, and without disturbing them, we can use them as sentinels for new pathogens. So they are our little filters to find relevant things. And so that works well. But sometimes we also find that the pathogen is actually not from the environment, but that we are the source of the pathogen, which is perhaps less interesting from an anthropocentric point of view. But from a conservation point of view, this was one of the most relevant things we ever found, I think, because that led to hygiene measures, which were then implemented in all field sites around Africa, at least those I know, to reduce spillover from humans to the animals. They are very important reservoir these days. We carry lots of pathogens to the last parts of the world. So this is a win-win for conservation, for primatology, and for infectious disease research more from an anthropocentric point of view. That's why I like these animals so much.
Another specific aspect of your project is that it's a long-term surveillance project. You spent now 24 years on it with considerable effort and found, like you said, quite a number of pathogens out there. For surveillance and possible prevention, do we need more of these inevitably more expensive and more labor-intensive surveillance projects on other species as well? I know that virologists are particularly concerned about bats. You also mentioned rodents earlier on. So if we really take One Health seriously and look at zoonotic events, do we have to start more of these long-term surveillance projects on a larger number of species and ecosystems?
Yes, so there are two very important points on here. I think we need to understand trends and shifts, and this is where you need long-term projects. And it's not like you collect samples for 25 years and then you start doing your research. You do research all the way long. You will find relevant things and publish things and so on. So I think building on long-term research is very essential and often still overlooked. There are great ecology studies or primatology studies or animal behavior studies, but they're not linked to people looking at these opportunities also from an infectious disease point of view. So to have more of what we did for Thai, and now we're doing it also for gorillas and chimps in Gabon, and for bonobos in DRC, and for gorillas in Central African Republic, for example. So with great aid, we have started that now already in a systematic way. But to have that on a broader scale would make sense and be a recommendation to many colleagues out there. That's one thing. And now with the new Helmholtz Institute here, I had the opportunity together with my team and the founding partners to create a concept for the whole institute. And I said, let's take those nice bits and pieces we have in the Thai region, in Ivory Coast. Maybe have the long-term data on the great apes. So that's already a cohort study, right? That's out there. Mm-hmm. But we have the small mammal work and the work we did on people were like more than classic 3 to 5 year projects funded from different sources. Let's try to create something we call a One Health surveillance, which is a really comprehensive surveillance where we look simultaneously at humans in cohorts. We look at humans in the clinics, in the little village hospital where people go when they're really sick. Have a cohort like people from the village living in and with that environment. But we also have everything that surrounds them, from livestock to the small mammals to the mosquitoes. We have climate data with microclimate loggers and bigger weather stations to climate longitudinally. And then in the same area, very close by, we have our great ape cohort since 25 years, right? So we can link that up. And this kind of deep dive in a very specific region is like 6 to 8 villages maximum, plus forest around. If we had more of these kind of weather stations for One Health around the world, this would be wonderful. And to get copied, most scientists don't want to be copied, but I think my biggest success would be if that concept of a One Health surveillance would get copied as much as possible. And that would be very helpful, because then you pick up signals with different climate change, with habitat change, with shifts in vector composition or reservoir compositions or hunting behavior by people. They shift the hunting because larger game is hunted out. What happens then? How does that alter disease risk for people? These are all things we can only understand if we do that longitudinally on many species and the environment.
Mm-hmm. When you're out there and looking for pathogens and diagnosing How do you see the impact of these new technologies coming in? This single-cell sequencing, the new sequencing technologies, I think particularly the nanopore sequencing, which allows sequencing in the field, but also new methods in serological analysis, antibody analysis. Do you see how this makes your work easier to find microorganisms generally and potential pathogens out there?
I think that the techniques which have been developed and are currently really muchly being all over the world are amazing and help us already now a lot. Since several years in our field sites, we have benign sequencing on site. If we have a little PCR on site, you do a generic PCR for filovirus or poxvirus or whatever, because you have to consider there will be new variants. It's never what you expect, so you need to be broad. But then what does that result mean? And that's where you need the benign. And you cannot wait until someone from your team or travel to your African. We have very strong collaborations with African partner labs, but do the confirmation over there, or in worst case, bring it all the way to Germany to do the sequencing. That doesn't make sense, especially if you want to be also on the alert side, right? And be able to inform communities and local authorities about, let's say, Ebola case in wildlife or anthrax case. We need to know that right away and not 3 months later. Mm-hmm. So these techniques help us a lot for rapid response on site, but they're also with the broader techniques like the multiplex serology, Luminex DSAs, and so on. They help a lot to get real signals about past exposure, also hinting towards things which are unknown so far, variants which you don't have the exact hit, but you know there will be something in between and that you can look specifically for that. So those techniques are very helpful. And the aim is, with this One Health Surveillance, this project which we are building at the moment, to bring as much as possible of that technique into the remote rainforest, into our communities, to do on the fresh material, to apply as much of these techniques, as many of these techniques as possible. Mm-hmm. And that always, just to say that here again very clearly, we are not doing colonial style research. So it's always with our African partners, our African partner labs. So it's really important that these in-country capacities are strengthened a lot. So, but the techniques itself, they're easy. They're cool.
How difficult is it to bring this fairly cutting-edge, sophisticated technologies to these remote areas? And another aspect that you also mentioned in the article is to train and work with your African colleagues there who do not necessarily have the same resources as a Western lab at hand.
So technically, I mean, there's strict regulations about export of any lab material, and even a swab you would have to declare to the German customs, and takes lots of time and effort, but you have to do it, and it's good we have these rules. And so that's just something to consider. And then on the African side, depending on the country, you also have certain rules which always follow. And then the techniques themselves, like the midnight sequencer, many people may have seen one, but they're more like a bigger USB stick, and you can put it in your pocket and bring it to the field site. It's really very easy. So there are nice developments to be very mobile with these things. And then the question is of the training, and the training of our collaborators on site. That's also something where I'm a very big promoter of long-term collaboration, long-term projects together. You can do a 2-year project here and there, but you will not leave many traces, that things will break down depending a bit on the context. Yeah. So we have these long-term partnerships, and that's really a family. They're colleagues. We discuss, the future ideas. We have all these ideas in the drawer. When there's a funding opportunity, we just open the right drawer and pull out the idea, which we developed already, Mm-hmm. And so that's a very trustful relationship and very respectful, and that's very important. And within that, we have African PhD students and postdocs. And I'm a big fan of doing capacity building by doing concrete research, not just a 2-week course, but to really get the routine. And this is what we have done over the past years, and we continue to do. And now our first PhD students, they are now our country coordinators in Ivory Coast, for example, right? And they become getting important positions, and they push their community. So that's very important. And they also like to come here from time to time. If it's not in the winter season, then it's good to have them here for a few months with their own samples. They learn this the techniques here. They go back to the country and they apply in their labs, sometimes supported by staff from here. So it's really a lot of exchange, come and go. You will always find someone from our partners here or someone from us over there. So that's a nice mix and nice family.
In the LADA context, as you said earlier, that you hope that people will copy you and set up these long-term surveillance projects all over the world. Should then the rich countries be much more supportive for the local scientists and for the local communities or research institutions there, since this is really a huge health problem, eventually, as COVID and Ebola have shown. If we decide that we have more surveillance for our own sake, how and who should then get into this and provide the funding and the infrastructures and this? This is your project, but you said we probably need this on a larger scale and on more sites across the world.
Yeah, I think we all have seen that infectious disease don't really care about borders and distances anymore with the activity we have. And we also know that many of the habitat changes we see, especially in those so-called high-risk areas like the tropics, where you have, ecological changes, but you also have very weak health infrastructure. You have often almost no veterinary infrastructure. So you have this dangerous, and you have high mobility these days and population growth. So you have a dangerous mix. We cannot say it's their problem. They should look after their health and the disease risk in their country, because we also do it nicely here in the Greifswald region, and we're very good at that. Because there is a reason why many of the countries of the world are not doing so well, and we are part of that reason if you look into the history. So given that, I think pandemic prevention preparedness is a global responsibility. That's where we should really work all together. And there are some funding mechanisms now on the larger scale, and I hope they will have effect. We will see they just started with pandemic fund and so on. But I think what's very valuable is to strengthen individual partnerships or network or smaller networks, because they will be sustainable. And there, I see other colleagues also have built up these kind of long-term relationships and so on. I think that's something which needs the support. And if the money comes mainly from the north, that's totally fine. Because, as I said, it's a global responsibility to just not mean that we dominate the research down there. It should be done mainly by people who are there on site and who will see the signals hopefully early and detect to extinguish those fires before they become too big, as we saw in West Africa with Ebola.
How do you see the possibilities and the chances if you train your local communities, your local scientists, your local researchers and veterinarians and boarders? Do you see that this could also have an effect there to decrease the risk and help to prevent zoonotic events from happening in the first place?
Yes. So there are different scales here. One is sort of the national, the countrywide scales. And I'm very happy to see that many of our partner countries have now a One Health strategy for the whole country. And they have like a One Health Secretariat, which is in some cases even situated above the ministries. So you don't have a competency problem. Yeah. Actually, Germany doesn't have a national One Health strategy, by the way. So they're really advanced. Now the question is the implementation. And that's really, you have to strengthen on the broad scale, you have to strengthen public health, and you have to strengthen veterinary health, and also the environmental monitoring. And those are huge fields. And you have to be careful that you keep an eye on the gradients you have within the countries. I see that a lot of research and capacities is built up in the capitals and the places where people have studied, where the new middle upper class colleagues are living, and the willingness for example, a medical doctor to go into a village and work there with the communities and be in that small hospital is small, because they can have a, very modern life in the city. And so you have gradients in the countries, and we have to make sure that the villages and the rural areas are not forgotten, because those are the areas where you have the closest human animal environmental impacts. So it should almost be, the other way around, but it's not, for understandable reasons. So strengthening the countrywide surveillance systems is important. That's also linked to better education systems, right? I mean, university education varies a lot from topic to topic and university to university. We know that phenomenon from Germany as well, but it's much more severe in many of the countries. Right? So you're really at the roots of the problems there. However, with projects like we do, we at least train the next generation as well of trainers. We sharpen the eyes of the local doctor to see and to report and to know how to report and have the channels to report the unknown, like something real. Suddenly 10 people show a symptom he normally does not see very often, or, has never seen, or things like that, which normally tend to drown. And so to sensitize people from on the side, basically to the universities is something we need to do. So I know it sounds a big challenge, and the little Helmholtz Institute for One Health will not solve all of that, but we create examples and have a locally needed.
Thank you very much for your podcast and your answers and your time.